This lecture will cover many of the basic topics in molecular biology, providing an all-in-one review for those needing to come back to this topic, or a brief overview for those new to the subject. The material will be delivered in a manner that I believe is suitable to a wide range of audiences, from undergrad to graduate levels, for academic researchers, medical students, residents, and anyone interested in this topic. We will cover the concepts behind the central dogma, that is, DNA to RNA to protein, and touch on some of the post-translational and epigenetic changes that occur. I will briefly cover some of the genetic terminology and basic concepts of inheritance, with the intention of doing a more thorough lecture on genetics in the future. And finally, I will go over some of the nomenclature rules so you can understand the molecular shorthand that is often used in clinical reports. There is an abundance of material here, and you probably don't want to sit through content you are already familiar with. So if you would like to skip ahead to a subject, feel free to click on the timestamps for the topics listed in the description below. The human genome has about 6 billion base pairs, or more specifically 3.2 billion per haploid genome. Of this massive DNA library, only about 25,000 genes are present in the human genome, accounting for about 1% of the total DNA sequence. So the vast majority of our DNA doesn't even code for proteins. Compare that to a worm like the nematode, which has about 20,000 genes, or a tomato that has more genes than us, at about 32,000. So what gives? We've learned that it isn't about how many genes in our genome that matters. It's how the genes are transcribed, modified, translated, and the interdependence of genes on each other. So even though we only have about 25,000 genes, we actually have far more proteins, with estimates of over 1 million proteins. This complexity is not entirely shared with the tomato. There are a couple terms introduced in this graphic that I would like to go over for completeness. We use the term genome to refer to the entire genetic material in a cell, or the entire DNA sequence. Transcription is the process of creating messenger RNA from this DNA sequence, and the transcriptome refers to the entire messenger RNA content. Messenger RNA, or mRNA, is what eventually gives rise to protein, and by extension, the proteome, or the entire protein content. More on these components later. So again, changes at the transcriptional and mRNA levels increase the size of the transcriptome relative to the genome, and the different post-translational modifications exponentially increase the complexity of the proteome relative to the transcriptome. The central dogma of molecular biology states that DNA makes RNA makes protein. But this only describes the 1% of our genome that codes for proteins. The remaining 99% of non-coding DNA, which we arrogantly use to refer to as junk DNA, contains many important regulatory sequences. These include promoter regions that provide a binding site for the transcription machinery, typically found just ahead of a gene on the DNA strand. Enhancers, which provide binding sites for proteins to help activate transcription and can be found before or after the gene they control, sometimes extremely far away from the gene they affect. Silencers are DNA sequences that repress transcription, and like enhancers can be found before or after the gene. Other regions of non-coding DNA provide instructions for the formation of special kinds of RNA molecules, like transfer RNAs and ribosomal RNAs that help assemble proteins, or microRNAs that block protein production, and other special types of RNAs. Some structural elements of chromosomes are also part of non-coding DNA, such as the telomeres at the ends of chromosomes that protect them from degradation after replication, or the centromere, which is the constriction point of the X-shaped chromosome pair. Other regulatory elements and functional regions in non-coding DNA have yet to be discovered, and researchers are working to understand the location and role of these genetic components. Let us start with the building blocks of DNA and RNA. These are the nucleic acids, which are defined by their bases. DNA bases include adenine, thymine, cytosine, and guanine, abbreviated as A, T, C, and G. RNA has the same bases except uracil replaces thymine. A nucleoside describes the base attached to a deoxyribose or ribose sugar. A nucleotide describes the base, the sugar, and the attached phosphate group or groups. The purines adenine and guanine have 2 carbon-nitrogen rings. The pyrimidines cytosine and thymine have 1 carbon-nitrogen ring. Note that cytosine can convert to uracil through the process of oxidative deamination, whereby cytosine loses the 4' amino group in exchange for an oxygen. Other than the obvious implication of introducing a point mutation, this process can actually become an issue with old tissue samples received for NGS studies or other PCR-based studies. And this includes FFPE samples, or formalin-fixed, paraffin-embedded tissue. Typically, after 5 years, a sample will show significant C to T transitions, and this gets really bad in samples older than 10 years. The same conversion can occur with deamination of adenine, resulting in a guanine. The phosphate group of each nucleotide is covalently bonded to the phosphate group of the adjacent nucleotide, forming the outer backbone of the nucleotide strand. The polymerization of the nucleotide strand loses 2 of the 3 phosphate groups, providing enough energy to catalyze this polymerization. The 5' carbon of one nucleotide is then covalently bonded to the 3' carbon of the adjacent nucleotide, eventually forming a single strand of DNA. A double strand is the result of base pairing between A and T, or C and G. This pairing relies on hydrogen bonds. AT pairs have 2 hydrogen bonds, while CG pairs with 3 hydrogen bonds. This difference results in a slightly stronger bond between CG pairs, which has consequences in certain lab tests, such as primer design for PCR analysis or melting curve analysis. This base pairing is facilitated by the presence of water. The bases are hydrophobic, while the phosphate sugars are hydrophilic. In the case of DNA, base pairs stack on the inside of the double strands, and a secondary structure forms, whereby the 2 strands twist, forming a right-handed helix, having 10 bases per turn. For RNA strands, which tend to be a single strand of much shorter length, a complex 3-dimensional structure is formed. The 3' and 5' ends of the strand, designated by the carbon position of the phosphate group at one end, provides directionality for the nucleotide strands. The 2 strands align in opposing directions, such that the 3' end of one strand is paired to the 5' end of the other strand, as seen in the picture here. This results in a sense and anti-sense strand, or parallel and anti-parallel strand. This directionality is important for the function of DNA and RNA. A helpful way to remember directionality is the phrase read up, synthesize down. That is, the DNA template is read in the 3' to 5' direction, while synthesis of DNA, RNA, and proteins occurs in the 5' to 3' direction. So while the transcription complex reads DNA in a 3' to 5' direction, the mRNA is being synthesized 5' to 3'. In other words, the template DNA strand and RNA strand are anti-parallel. When translation occurs, the ribosome moves along the mRNA transcript 5' to 3', synthesizing proteins in the 5' to 3' direction. This mRNA intermediate results in a protein chain being created in the same direction as the original DNA template strand. We will discuss the details of these steps shortly. How do we fit approximately 2 meters of DNA into a cell nucleus that's only 6 microns in diameter? By packing the DNA into chromatin and chromatin into chromosomes. Histones are proteins that complex together to help package the DNA into what we call nucleosomes. And these proteins are the main component of chromatin. There are 5 histone proteins: H1, H2A, H2B, H3, and H4. H2A, H2B, H3, and H4 come together to form an octamer, or 8 proteins complex together, with H1 known as a linker histone. The negatively charged DNA strands wind around the positively charged histone octamer about 1.7 times around, or approximately 147 base pairs in length. This DNA histone structure is called the nucleosome. Nucleosomes then stack together, resulting in a supercoiling of the DNA. It is this supercoiling that allows the full length of the human genome to fit inside the nucleus of a cell. Modifications on histone proteins allow for the chromosome to be wound up tightly, which we call heterochromatin, or loosely, which we call euchromatin. And this has ramifications on gene expression in those segments of DNA. Normal human cells contain 23 chromosomes, each in duplicate. One copy is inherited from father, the other from mother. Among the 23 chromosomes are the sex chromosomes, XX for female and XY for male. A centromere is present in each chromosome that allows microtubules to attach and direct separation during cell division. The centromere is located off-center and results in a long and short arm for each chromosome. Telomeres are repeat sequences at the end of each chromosome that protects the DNA. After every set of replication, some small fraction of DNA is lost, and this is taken from the telomeres. This is why aging, or multiple successive cell divisions, is associated with shorter telomeres. The structure of chromosomes is very complex when unwound, as would be found in a G1 or G0 resting state, or what we call interphase. But we often simplify chromosomes into their compact configuration, as would be found in a cell undergoing mitosis, as demonstrated here in this cartoon. The long arm we designate Q, the short arm is P. Remember P for petite. The arms are separated by a centromere, and the ends of the chromosomes hold the telomeres, which are both non-coding repeats of DNA. The location of the centromere on the chromosome determines what grouping it will fall into: metacentric, submetacentric, and acrocentric chromosome. The term ploidy defines the number of chromosomes. Haploid is 1N, or one copy of a chromosome. Normal is diploid, 2N. Aneuploidy means there is an abnormal number and is not specific. Mosaicism describes more than one genomic sequence derived from a single origin and coexisting within a single organism. Chimerism describes more than one genomic sequence within an organism that have completely different genetic origins. So the difference between mosaicism and chimerism can be subtle, but has to do with the origin of the genomic material. When a cell is ready to divide, it has to make a second copy of all its DNA. Each of the 2 strands serves as a template for a new strand. Therefore, replication is described as a semi-conservative process, meaning that each DNA double helix will have one strand from the parent DNA and one strand synthesized from scratch. Because DNA can only be synthesized in the 5' to 3' direction, each strand has a slightly different replication process. The leading strand of DNA is the 3' strand. allowing the synthesized DNA to form 5' to 3' uninterrupted. The lagging strand is the 5' strand, which has multiple interrupted segments of DNA that are later connected with ligase. If this does not make complete sense now, hold on a little longer and I will show you an animation of this process. DNA replication relies on a replication complex that includes several proteins. Some of these proteins include helicase to unwind the DNA, polymerase which synthesizes the new DNA strand, ligases to glue interrupted strands or Okazami fragments together, and primases to help initiate the polymerase enzyme. Several other proteins that are important for DNA replication, though not directly part of the replication complex, are also present. Some examples of these include topoisomerases to add or reduce the supercoiling of DNA strands, particularly important in prokaryotes where DNA is circular rather than linear, and telomerases, unique to eukaryotes with linear DNA, which are vital to maintaining the genetic material at the ends of the linear DNA strands. It is helpful to know that different polymerases exist. Alpha and delta replicate nuclear DNA. Beta and epsilon are responsible for DNA repair, and gamma polymerase replicates mitochondrial DNA. Polymerase accuracies vary from one type to another. Although errors can occur, the cells have intrinsic mechanisms for repair to address these errors. Tumor cells may lose this repair ability, such as in the case of microsatellite instability or MSI tumors. We will discuss microsatellite instability in more detail in Part 2. To help solidify the process of DNA replication, I think it is helpful to see it from start to end, and so I would like to show you this wonderful animation from DNAi.org that does a fantastic job summarizing this process.
During DNA replication, both strands of the double helix act as templates for the formation of new DNA molecules. Copying occurs at a localized region called the replication fork, which is a Y-shaped structure where new DNA strands are synthesized by a multi-enzyme complex. Here, the DNA to be copied enters the complex from the left. One new strand is leaving at the top of frame, and the other new strand is leaving at the bottom. The first step in DNA replication is the separation of the two strands by an enzyme called helicase. This spins the incoming DNA to unravel it at 10,000 RPM in the case of bacterial systems. The separated strands are called 3' and 5', distinguished by the direction in which their component nucleotides join up. The 3' DNA strand, also known as the leading strand, is diverted to a DNA polymerase and is used as a continuous template for the synthesis of the first daughter DNA helix. The other half of the DNA double helix, known as the lagging strand, has the opposite 3' to 5' orientation and consequently requires a more complicated copying mechanism. As it emerges from the helicase, the lagging strand is organized into sections called Okazaki fragments. These are then presented to a second DNA polymerase enzyme in the preferred 5' to 3' orientation. These sections are then effectively synthesized backwards. When the copying is complete, the finished section is released and the next loop is drawn back for replication. Intricate as this mechanism appears, numerous components have been deliberately left out to avoid complete confusion. The exposed strands of single DNA are covered by protective binding proteins, and in some systems, multiple Okazaki fragments may be present.
As a reminder, DNA replication is only one part of the cell cycle, designated as the S phase. The S phase is followed by G2, where DNA repair and the preparation for cell division occurs. This is followed by cell division, progressing through the 4 phases of mitosis and ultimately ending with 2 daughter cells in G1 or G0 if terminally differentiated. Certain checkpoints are built into this replication cycle to make sure the process of DNA replication and cell division progress appropriately. If a cell fails these checkpoints, an attempt is made to correct the error, or if the error cannot be corrected, the cell undergoes apoptosis. Defects in proteins that regulate the cell cycle at these checkpoints create opportunities for defective cells to grow when they shouldn't, often leading to a tumor. Our cells have mechanisms in place to repair DNA damage to minimize potentially deadly consequences associated with that damage. The source of damage can be exogenous, like UV radiation or exposure to carcinogens, or it can be from endogenous sources, such as replication errors or exposure to reactive oxygen species. Single-stranded DNA breaks are the most common lesion arising in cells. Direct reversal is a chemical correction mechanism that reverses any base switches or alterations in the chemical structure of that base. This mechanism does not rely on a template and does not affect the covalent bonds of the DNA backbone. The protein methylguanine methyltransferase, or MGMT, is one example of a direct reversal repair protein that removes aberrant methyl groups on guanine bases. MGMT may sound familiar to some of you, as it has significance in glioblastoma therapy. Briefly, in some brain tumors, hypermethylation of the MGMT promoter region prevents this protein's expression, which makes the tumor more susceptible to alkylating chemotherapy agents, since it has lost this vital repair mechanism. We will discuss methylation further in the epigenetics section of this lecture. Another repair mechanism is nucleotide excision repair. In the presence of UV light, pyrimidine dimers form, resulting in a kink in the DNA code. This group of repair enzymes excise the defect and resynthesize the DNA. Nucleotide excision repair is defective in xeroderma pigmentosa. Base excision repair removes a single base from its deoxyribose sugar and replaces it with the correct base. Mismatch repair systems, or MMR enzymes, represent a collection of enzymes that work in pairs to detect areas of the code that do not match the template DNA and repair that sequence. These enzymes are implicated in Lynch syndrome, as we will discuss in the next lecture when we discuss microsatellite instability. Double-strand breaks occur through high-energy damage. Think radiation energy, for example. These are very serious breaks, as large portions of chromosomes can be lost or rearranged if not repaired. There are two main mechanisms for repair in double-stranded breaks: non-homologous end joining and homologous recombination. Non-homologous end joining uses DNA ligase 4 complexed with XRCC4 to join two ends together. The complex relies on overhanging sequences as a short template to match the two ends together. This is an important mechanism in B and T cells during VDJ recombination, where double-stranded breaks occur as part of the lymphocyte's natural development. Homologous recombination requires the presence of an identical or nearly identical sequence to be used as a template for repair of the break, and because of this is more accurate than non-homologous end joining. Before we discuss transcription, let us first take a look at the structure of a gene. This diagram is a simplified depiction of one strand of DNA containing a representative gene. The 5' end is on the left and the 3' end is on the right. The enhancer and silencer for this gene are outside of view. The transcription start site is given the +1 designation. Everything 5' from this is considered upstream, and everything 3' from this is downstream. A promoter region is located upstream of the coding region and is the area where the transcription complex assembles. Transcription of the mRNA sequence begins from the initiation site until it reaches the termination region. The DNA sequence between the transcription initiation site and the termination site is the coding region. This coding region of the gene contains exons and introns, which we will discuss momentarily. If the direction of this diagram is confusing to you, that's because you are missing one key bit of information. Remember I said that transcription occurs in a 3' to 5' direction. But what we are looking at here is a 5' to 3' structure of a gene. In fact, what we are looking at is the coding strand of DNA, whose sequence corresponds exactly to the mRNA transcript that will be produced. The complement strand of this gene, or the antisense strand, is referred to as the template strand. The template strand is a non-coding strand that is transcribed by the RNA polymerase in a 3' to 5' direction. By transcribing the antisense strand of a gene, the resulting mRNA sequence will match exactly the coding sequence, or the sense strand of this gene. And that is why genes are depicted 5' to 3'. Again, transcription describes the process of copying a gene into messenger RNA, or mRNA, and this is a highly regulated process. Various transcription factors come together to form a transcription complex at the promoter region, including RNA polymerase. These upstream promoter regions comprise of TA-TA repeats, or CAAT sequences that signal transcription complexes to bind. Other proteins in the transcription complex interact with regulatory sequences in the DNA, such as the enhancers and silencers, to affect gene transcription. Enhancers and silencers may be located adjacent to a gene, or sometimes on a different chromosome even. And just like DNA, there are different types of RNA polymerases out there. RNA polymerase I transcribes ribosomal RNA. RNA polymerase II transcribes messenger RNA, as well as microRNA, small nuclear RNA, and other RNA sequences. And RNA polymerase III transcribes transfer RNAs. Of course, a gene is not transcribed once, then left alone. Rather, the gene that is open to transcription will be transcribed multiple times, until a signal from the cell stops it from happening. This image depicts a single strand of DNA being transcribed multiple times. The strand of DNA is at a diagonal in the center of this image. Multiple RNA polymerases are transcribing this portion of DNA already, each producing their own strand of RNA, coming off like bristles in a wire brush. The young strands of RNA are located on the bottom left, the shorter strands, with the largest RNA strands at the top right representing nearly complete transcripts. Now let's see the process of transcription play out in the following animation.
The central dogma of molecular biology, DNA makes RNA, makes protein. Here the process begins. Transcription factors assemble at a specific promoter region along the DNA. The length of DNA following the promoter is a gene, and it contains the recipe for a protein. A mediator protein complex arrives carrying the enzyme Rna Polymerase. It maneuvers the RNA polymerase into place, inserting it with the help of other factors between the strands of the DNA double helix. The assembled collection of all these factors is referred to as the transcription initiation complex, and now it is ready to be activated. The initiation complex requires contact with activator proteins, which bind to specific sequences of DNA known as enhancer regions. These regions may be thousands of base pairs distant from the start of the gene. Contact between the activator proteins and the initiation complex releases the copying mechanism. The RNA polymerase unzips a small portion of the DNA helix, exposing the bases on each strand. Only one of the strands is copied. It acts as a template for the synthesis of an RNA molecule, which is assembled one subunit at a time by matching the DNA letter code on the template strand. The subunits can be seen here entering the enzyme through its intake hole, and they are joined together to form the long messenger RNA chain snaking out of the top.
Transcription continues until the RNA polymerase reaches a termination sequence in the DNA. Once the transcription process is complete, mRNA needs to be processed to exit the nucleus and the sequence formatted in a way that allows it to be translated into protein. These modifications include, first, splicing out the introns from the sequence. Remember that exons get expressed. How these introns are spliced can alter the mRNA sequence, for good or bad, and we'll discuss splicing in a minute. The second post-transcription modification is to place a cap on the 5' end to protect the mRNA transcript from exonuclease degradation. Exonuclease degradation is a key defense mechanism against foreign RNA which lack the 5' cap. This cap also facilitates transport of the mRNA out of the nucleus and into the cytoplasm, where it can be translated into protein. And lastly, the addition of a 3' polyadenylated tail helps protect the other end of the mRNA and enhance ribosomal activation and recognition of the mRNA sequence. Let's briefly discuss splicing out introns. Introns are non-coding sequences about 10-fold longer than the exons bordering them, and they need to be excised from the mRNA transcript before a viable protein can be made. Introns contain small conserved sequences known as splice site donors and splice site acceptors. The RNA transcript loops on itself so that a splice donor meets with a splice acceptor, forming a spliceosome that eventually cleaves the intron out and ligates the two bordering exons together. Now, imagine a mutation occurs in one of these conserved sequences. If either a donor or acceptor site is mutated, the intron carries on to the final mRNA transcript and gets translated into the protein sequence, likely affecting the protein function. Alternatively, if a mutation in an exon results in a donor or acceptor site sequence, then a portion of that exon may be spliced out that should not have been, possibly resulting in a defective protein. Introns are not completely useless segments of code. In fact, they are vital to the proper function of the cell. The type of intron we just discussed are called ribozymes. A ribozyme is an RNA molecule that has the ability to catalyze a biochemical reaction, such as RNA splicing. Other types of introns are functional nuclear introns that are important for the expression of the gene on which they reside. This is not a feature present in all genes, but has been described in some. There are also cases in which introns contain genes within their sequence that code for small nuclear RNA, which are important for the translation of messenger RNA. Introns are also important for alternative splicing. Alternative splicing is a naturally occurring, well-regulated process that allows a single gene to encode multiple proteins with similar domains. A domain describes a functional unit of a protein. The most common form of alternative splicing is exon skipping, where certain exons are left out of the final transcript, as depicted here. Proteins A, B, and C are all protein isoforms derived from the same gene. An example of alternative spliced proteins are hormones. There are other processes and configurations of alternative splicing, but I will leave those for you to look up if you are interested. So, to review, a gene codes for a messenger RNA. The mRNA is processed and then is ready to be translated into a protein. Translation is the conversion of the mRNA sequence into a protein and is based on a trinucleotide codon. Ribosomes recognize the 5' cap of the mRNA and read along until they get to the start codon, typically an AUG sequence, which codes for the methionine amino acid. The ribosome then begins the translation process, moving along the mRNA one trinucleotide codon at a time, adding one amino acid to the protein chain per codon, until it reaches the stop codon. Transfer RNAs, or tRNAs, are responsible for carrying amino acids to the ribosomal complex. If the tRNA matches the codon of the sequence, it releases its amino acid to be bound to the growing peptide chain. Codons are specific to the amino acid, with the third base in a codon being a wildcard, known as the wobble position. This is possible because we have 64 possible codons and only 21 amino acids to work with, leaving some redundancy in the code. The start codon in eukaryotes always codes for methionine, and there are 3 stop codons, UAA, UAG, and UGA. This has to do, at least partially, with the number of hydrogen bonds in the sequence, allowing the ribosome to detach more freely. Depicted here are 2 common representations of the codon translation. I prefer the wheel on the left, as it displays more information than can be gleaned from the table, but I have included both for your reference. The astute among you may notice something off about this particular codon wheel. This wheel is the one commonly published in textbooks and online, which shows you the first codon in the center, the second around that, and the third codon position or wobble position at the edge, to give you the same information from the table. It's useful if you want a direct translation of the codon sequence and nothing else. This one, on the other hand, emphasizes the importance of the second codon. I did not create this wheel. This is the work of Milton Sayer at UCSD, with a link in the references section for those that wish to read his paper on this topic. It turns out that position 2, or P2, specifies the type or group of amino acid. P1 usually specifies the specific amino acid, and P3 is highly redundant, as we just discussed, with several bases specifying a particular amino acid. When P2 is a T, or uracil in the case of RNA, all amino acids are strongly hydrophobic, without exception. If P2 is an adenine, all amino acids are strongly hydrophilic, also without exception. When cytosine is in position 2, codons code for semipolar amino acids. The same is true for guanine at P2, mostly semipolar amino acids, with 2 exceptions: arginine, a strongly hydrophilic residue, and the chain termination codon opal, or UGA. For more interesting associations of the codon table and wheel, I would recommend this paper in the references. Displayed here are the amino acids grouped by their hydropic relation on the left, or their overall charge on the right. Amino acids are named by the molecular structure of their side chain, or R group, which provides the amino acid with its chemical attributes. For every amino acid, there is one amino group and one carboxyl group, both attached to an alpha carbon. The amino acid side chain also attaches to this alpha carbon. As the protein chain is being made, a condensation reaction occurs. The carboxyl group on one amino acid reacts with the amino group of a second amino acid, releasing one water molecule and forming a carbon-nitrogen peptide bond between the 2 amino acids. The amino and carboxyl groups of the peptide chain, in effect, provide directionality to the protein sequence. By convention, protein sequences are written in the same direction that translation occurs, from the end with the free amino group, or the N terminus, to the end with the free carboxyl group, or the C terminus. The resulting product of translation is the polypeptide strand, also known as the primary structure. The various chemical properties of the amino acids within this protein sequence interact with each other to form two basic motifs, alpha helices and beta pleated sheets. These motifs make up the secondary structure of the protein. The amount and configuration of the alpha helices and beta pleated sheets make up the tertiary or final protein form. From here, proteins may interact with other proteins to form complexes or quaternary structures. We briefly mentioned the term protein domain in one of the previous slides. A protein domain is a conserved part of a given protein sequence and structure that can function independently from the rest of the protein chain. Each domain forms a compact three-dimensional structure that often can be independently stable and retain its shape and function. Many proteins consist of multiple domains, each with a specific function and purpose, and one domain may appear in a variety of different proteins. Take, for example, the epidermal growth factor receptor protein, or EGFR. It contains an extracellular binding domain that binds a ligand, a short transmembrane sequence to fix itself to the cell membrane, and a tyrosine kinase domain responsible for phosphorylating tyrosine residues on other proteins in the cell's cytoplasm. Now let's take a step back and take a look at translation from the beginning.
The job of this mRNA is to carry the gene's message from the DNA out of the nucleus to a ribosome for production of the particular protein that this gene codes for. There can be several million ribosomes in a typical eukaryotic cell. These complex catalytic machines use the mRNA copy of the genetic information to assemble amino acid building blocks into the three-dimensional proteins that are essential for life. Let's see how it works. The ribosome is composed of one large and one small subunit that assemble around the messenger RNA, which then passes through the ribosome like a computer tape. The amino acid building blocks, that's the small glowing red molecules, are carried into the ribosome attached to specific transfer RNAs. That's the larger green molecules also referred to as tRNA. The small subunit of the ribosome positions the mRNA so that it can be read in groups of 3 letters known as a codon. Each codon on the mRNA matches a corresponding anticodon on the base of a transfer RNA molecule. The larger subunit of the ribosome removes each amino acid and joins it onto the growing protein chain. As the mRNA is ratcheted through the ribosome, the mRNA sequence is translated into an amino acid sequence. There are 3 locations inside the ribosome, designated the A site, the P site, and the E site. The addition of each amino acid is a 3-step cycle. First, the tRNA enters the ribosome at the A site and is tested for a codon-anticodon match with the mRNA. Next, provided there is a correct match, the tRNA is shifted to the P site, and the amino acid it carries is added to the end of the amino acid chain. The mRNA is also ratcheted on 3 nucleotides or 1 codon. Thirdly, the spent tRNA is moved to the E site and then ejected from the ribosome to be recycled. As the protein synthesis proceeds, the finished chain emerges from the ribosome. It folds up into a precise shape determined by the exact order of amino acids. Thus, the central dogma explains how the 4-letter DNA code is, quite literally, turned into flesh and blood.
Post-translational modifications are chemical modifications that are key to a functional protein. They regulate activity, localization, and interaction with other cellular molecules such as nucleic acids, lipids, and other proteins, and can occur at any time after the protein is made. These modifications occur at distinct amino acid side chains or peptide linkages and are most often mediated by enzymatic activity. These modifications include methylation, acetylation, phosphorylation, carboxylation, glycosylation, and hydroxylation. The enzymes that catalyze these reactions include kinases, phosphatases, transferases, and ligases, which add or remove functional groups, lipids, or sugars on amino acid side chains. There are also proteases, which cleave peptide bonds to remove specific sequences or regulatory subunits. Many proteins can also modify themselves using autocatalytic domains, such as autokinases and autoproteolytic domains. Some examples of these modifications include histone methylation and acetylation as a way of regulating chromatin state and ultimately transcription of a gene. You probably have heard of tyrosine kinases, which dimerize and autophosphorylate, another example. If you are interested in these modifications and you would like to review this subject further on your own, I will include links in the references. Epigenetics describes supragenetic modifications to the genome. These alterations do not change the genetic code, but rather influence how the code is read and interpreted by the cell. The two primary epigenetic changes are DNA methylation status and histone modifications. There are other types of modifications, but these are the big ones to know. Methylation typically occurs over CpG islands within the DNA, which are areas of the sequence where CG bases are plentiful. A lot of gene promoters have these CpG islands, and methylation of these regions results in repressed transcription, usually through steric hindrances or chromatin condensation. Methylation is a normal function of DNA regulation. It allows for genomic imprinting and X chromosome inactivation. Not all CpG islands are methylated, and the distribution of methylation is referred to as the methylation status. The methylation status of a cell type can vary between the different stages of tissue development. Stem cells show a predominantly unmethylated genome, but as the cell begins to differentiate, methylation increases. With regards to genetic imprinting, this describes the inherited pattern of methylation identified at certain alleles and loci within the genome. We know from imprinting disorders like Angelman's and Prader-Willi syndrome that the methylation status of alleles from maternal and paternal origins are different, and that difference is important. Aberrant methylation is associated with disease states and likely plays a role in all neoplasia. Take, for example, hypermethylation of the MLH1 promoter region, which is the most common cause of sporadic microsatellite instable colorectal cancers. Broader examples include hypomethylation of oncogenes or hypermethylation of tumor suppressor genes, for example. In DNA methylation, a methyl group is attached to a cytosine base at the 5' carbon by DNA methyltransferase, or DNMT enzymes. DNMT3A and 3B use a methyl donor called S-adenosylmethionine, or SAM. This methylation is maintained by DNMT1. Of course, there needs to be a way for the cell to remove this methyl group when it needs to, and for that, there are enzymes called TET, which stand for 1011 translocation methylcytosine dioxygenases. TET1, 2, and 3 are responsible for demethylating DNA in a multi-step process. It may be helpful to know that DNMT3 mutations are seen in certain myeloid neoplasms, like AML, or CHIP, which stands for clonal hematopoiesis of indeterminate potential. TET2 is often mutated in myeloid neoplasms as well, such as myelodysplastic syndrome, or MDS, as well as AML with poor prognosis, polycythemia vera, and CHIP, among others. Histone modifications are a type of post-translational modification that alters the chromosome structure. These are illustrations of some of the histone modifications that can occur, and barely scratches the surface of modifications that have been discovered. The location and type of modification are important. For example, methylation at one site of a histone can result in euchromatin, while methylation at a different site of the same histone can result in heterochromatin. The amount of methylation, methyl 2 versus methyl 3, can also alter the final outcomes and interfere with DNA binding proteins and transcription of the DNA. In general, acetylation decreases DNA histone interactions and improves accessibility of the DNA to transcriptional activation. In a nutshell, modifications to the histone proteins result in changes of the overall charge and configuration of that protein that either repels or attracts the negatively charged DNA. Genetics is the study of genes, genetic variation, and inheritance. We will be taking a very brief overview of basic genetics concepts and terms. I hope to expand on these topics and other medical genetic concepts at a later lecture. Broadly speaking, there is Mendelian genetics, which follow a set of strict laws that explain somewhat simple inheritance patterns, and Non-Mendelian genetics, which encompasses everything else that strays from those laws. Mendel's laws state, a parent who has two alleles for a gene can only pass on one allele or the other to each offspring. Two or more traits are inherited separately from each other and don't always occur together. And finally, a dominant allele will take charge over a recessive allele. The only way recessive alleles can be seen is if an individual is homozygous for the recessive allele. For clarification, an allele describes a variant of a gene. With every talk on genetics, there is at least some mention of the Hardy-Weinberg Equilibrium. This is a principle in population-based Mendelian genetics, stating that the genetic variation in a population will remain constant from one generation to the next in the absence of disturbing factors. Disturbing factors may include the presence of a mutation, non-random mating, small populations, and genetic drift, among others. If such strict conditions can be met, then the law predicts that both genotype and allele frequencies will remain constant because they are in equilibrium. The equilibrium formula is generated by the Punnett Square, an example of which is found at the bottom of this slide. The square takes into account the dominant and recessive alleles within a population. Dominant alleles are designated with a capital letter and recessive alleles with a lowercase letter. The frequency of the dominant allele is designated P, and the frequency of the recessive allele is Q. The genotype frequencies in a given population become the sum of outcomes, or P square plus 2PQ plus Q square, where P square designates the frequency of the homozygous dominant allele, Q square the frequency of the homozygous recessive allele, and 2PQ the frequency of the heterozygous alleles. If there are only two alleles at a locus, then P plus Q equals 1. It is possible to apply the Hardy-Weinberg formula to loci with more than two alleles, but for simplicity, we will only focus on one allele here. Let us put this into practice with an example. Let us assume that within a given population, we know that the frequency of the dominant allele, or capital A, is at least 20%. And individuals with this allele have brown eyes, a dominant trait. That means the recessive allele, which codes for blue eyes in this case, is present in 80%, since the sum of the frequencies P and Q must equal 1. To figure out the frequency of the genotypic combinations between these two alleles, we need to apply the Hardy-Weinberg equation. In this case, P squared is 0.04, Q squared is 0.64, and 2PQ is 0.32. If we were to draw out the Punnett square to scale, it would look something like this. The frequency of individuals with brown eyes, that is, expressing the dominant trait, would be the sum of all genotypes with the dominant allele. In this case, p square plus 2pq, or 36%. Those with blue eyes, or the recessive allele, represent 64%. Note that although brown is the dominant allele, it is expressed in a minority of people within this given population. This is because frequency and allele behavior are not the same, so do not assume that a larger frequency is automatically the dominant allele. Some useful terms to know: complete dominance is based on pure Mendelian genetics, where a trait is either dominant or recessive. However, as we know, this is not always 100% true. Incomplete dominance describes the blending of inherited phenotypes regardless of their dominance. Codominance describes both alleles being expressed without one masking the other. A great example of this is the ABO phenotype of red cells. Polygenic describes multiple genes that influence a particular trait. And along those lines, pleiotropic describes one gene that influences multiple phenotypes. Penetrance and expressivity are often confused, so let's review these two terms. Penetrance is used to describe whether or not there is an expression of the genotype in the individual. The genotype is present, but expression is either present or absent in this individual. This explains why some dominant traits are not expressed in certain individuals. Expressivity describes the differences observed in the phenotype between two individuals with the same genotype. It is the phenotypic spectrum of a given genotype. Within population genetics, certain patterns of inheritance exist. We can pictorially represent these patterns with pedigree charts, and we will see a few examples of these in a moment. These charts help us determine inheritance patterns for a given trait, and are broadly classified into autosomal, X-linked, and mitochondrial inheritance patterns. Some simple rules apply to interpreting these pedigrees. First, if a phenotype or trait exists in all generations, it is considered dominant. If it skips generations, it is recessive. Second, determining X-linked versus autosomal inheritance requires that we analyze male-to-male inheritance, or father-son inheritance. If a son inherits the trait from a father, then it is autosomal and cannot be X-linked, since fathers can only pass on their Y chromosomes to their sons. In X-linked dominant, the daughters may also show the trait. If X-linked recessive, then daughters may be carriers of the trait and expression will skip a generation. If only males are affected in several generations without skipping, you may be dealing with a Y inheritance pattern. Lastly, for mitochondrial inheritance, since mitochondria can only be inherited from mothers, this inheritance pattern will affect all offspring of a mother. The trait will terminate after an affected son, but continues in successive generations of an affected daughter. Now let's look at some examples. If you haven't seen these before, or need a refresher, allow me to orient you. Each row is a generation. Males are represented with squares, females are circles, and unknown individuals are represented by a diamond. Shaded individuals are considered affected, in that they express the phenotype or have the trait. Unshaded individuals are not affected. A half-shaded individual is a known carrier. The lines between individuals tells us their relationship to one another. Marriage lines connect from the side. Sibling lines connect from the top. In consanguineous marriages, a double line is used to connect from the side. Now, let's apply those rules that we just mentioned, and I encourage you to draw out the Punnett squares for each of these to help follow along. In autosomal dominant, both males and females will possess the trait or express the phenotype, and it will be present in every generation because it is dominant. Any offspring from a single affected parent will have 50% chance of inheriting the dominant trait. If both parents have the dominant trait, then the outcomes will depend on whether a parent is homozygous or heterozygous for that dominant trait. If both are heterozygous, then the risk of being affected is 75%. If at least one of the parents is homozygous dominant, then the risk is 100% in the offspring. Autosomal recessive traits will also affect men and women equally and can skip generations. Often, there is only a single generation affected, and we may not know that the parents were carriers. If both parents are carriers, then there is a 25% risk of an offspring being affected, and a 50% chance of having an offspring being a carrier. A common test question is to ask about the risk of having a carrier offspring versus a non-affected offspring being a carrier. The knee-jerk response is to say they are both 50%, which is actually incorrect. If we were to look at the risk of an unaffected offspring being a carrier, it would actually be 2/3. If this last point sounds confusing, draw out the Punnett Square. 1/4 of the offspring will be homozygous recessive, and therefore will display the phenotype or trait. That leaves 3/4 of the siblings not displaying the phenotype or trait, of which only 2 of those 3 has a chance of being a carrier. So the risk of having a carrier offspring may be 50%, but the risk of an unaffected offspring being a carrier is 2/3. If only one parent is a carrier, then there is also a 50% risk of being a carrier in the offspring, yet none of them will be affected since the other parent is not a carrier of the recessive gene. If one parent is affected, that is to say they are homozygous for the recessive trait, then all offspring become carriers. Again, if any of these scenarios sounded confusing, practice drawing out the possibilities on a square. X-linked dominant affects all generations, male and females alike, but again there is no male-to-male inheritance in an X-linked pattern. An affected female provides a 50% risk of having an affected offspring, since both boys and girls receive one X chromosome from their mother. If the mother is homozygous dominant for a trait, then all offspring become X-linked dominant for that trait. If a father carries an X-linked dominant trait, then all his daughters will become affected. In X-linked recessive, a father will have all his daughters become carriers. A heterozygous recessive mother provides a 50% risk of male offspring being affected, and 50% risk of female offspring becoming carriers. Homozygous recessive mothers will bump those percentages to 100%. Because mitochondria are inherited from mothers, there is only maternal transmission to all offspring, and therefore daughters are the only carriers. Mitochondrial diseases are varied and include subacute necrotizing encephalopathy, or Leigh syndrome, cardiomyopathy and conduction defects, and Pearson syndrome, among many others. Keep in mind that not all changes in the genetic code result in a change at the phenotypic level. A change of any type in the genetic code is called a variant. A variant is not synonymous with a mutation. When a variant occurs in less than 1% of the population, we may refer to it as a mutation. Mutations may be pathogenic or benign. So not all changes in the genome are mutations, and not all mutations are created equally. This is one reason why using the term variant is preferred over mutation. The type of change, its location, the frequency of the change within the population are all important factors in determining the significance of the variant. This is a great time to discuss single nucleotide polymorphisms, or SNPs. A SNP is a variant that occurs at a single specific nucleotide within a person's genome and is also found in greater than 1% of the population. Again, we generally consider variants that occur less frequently than 1% to be true mutations. If the SNP occurs in greater frequency among the population, it is less likely to be detrimental. However, keep in mind that SNPs are not always a benign finding, as in sickle cell disease or cystic fibrosis, where the point mutation in these diseases may be present in greater than 1% of a given population. By convention, the most common allele at each SNP locus is designated capital A, and the less common allele is designated capital B. Since there are 2 copies of each chromosome, the possible allelic combinations are AA, BB, and AB. These concepts will be useful in the second lecture when we discuss microarrays in disease states. Let's go over some useful definitions. Somatic variants are acquired, meaning they are not inherited, nor are they passed on to offspring. Germline variants you are born with. They are present in every cell in the body and can be passed on to the next generation. Loss of heterozygosity, or LOH, is the result of losing a segment of DNA from one chromosome, leaving only one copy of that DNA segment on the other chromosome. Uniparental disomy is a form of LOH that occurs when a person has 2 copies of a chromosome or part of a chromosome from one parent. Driver mutations describe cancer mutations that actively provides a selective growth advantage to the tumor. Passenger mutations describe cancer mutations that are present but don't seem to be required for tumorigenesis or cancer survival. Allele ratio and variant allele frequency are a couple terms you may come across in the diagnostic laboratory. The allele ratio is defined as the frequency of a mutated gene divided by the frequency of the wild-type gene. The variant allele frequency, or allelic fraction, is the frequency of mutated genes divided by the sum of the wild-type and mutated gene frequencies. In population genetics, allele frequency refers to the percentage of a population that carries that allele. You may hear this term used this way in reference to inherited diseases. In molecular diagnostics, allele frequency refers to the percentage of DNA in a specimen that carries a specified sequence, also known as allele burden, and can be a marker of residual disease in these specimens. An easy way to remember the difference between AR and VAF is that a ratio is one number divided by one number, while a frequency takes into account the entire population or all available possibilities in its denominator. We will discuss these and other examples in further detail when we talk about next-generation sequencing at a later date. Listed here are some of the various types of mutations. A missense mutation is a single DNA base pair change that results in a different amino acid than normal. If the substitution occurs in the wobble position of the codon, though, there may not be a change in the amino acid, given the high degree of redundancy in the wobble position. In these cases, we call it a silent mutation. Nonsense mutations are a single DNA base pair change that results in a premature stop of the translation process, resulting in a truncated protein. Insertions and deletions are self-explanatory. These may result in a frame shift if the inserted or deleted sequence is not divisible by 3. If they were divisible by 3, then the codon sequence remains intact and is only shifted without being disrupted. The subsequent protein domains in these cases of in-frame shifts are less likely to be affected. Duplications describe a sizable portion of DNA, often a gene, that is copied one or more times. Multiple duplications of an oncogene, for example, also called an amplification, are described in several cancers, such as the amplification of ERB2 in breast cancer. This is different from repeat expansions that are much smaller repeating sequences, usually 3 bases long and generally no more than 10 bases in length. Frameshift is the result of a duplication, deletion, or insertion that shifts the reading frame in transcription and translation, altering the sequence from that point on. A conversion is a replacement of a segment of DNA by a homologous sequence of DNA, resulting in 2 identical sequences on both DNA copies. Translocations describe rearrangements between different chromosomes and can be balanced or unbalanced. Balanced describes an even exchange of material with no extra or missing genetic information. This type of translocation can sometimes be fully functional as long as the translocation doesn't occur within a critical region, such as a gene. Unbalanced translocations describe the unequal exchange of chromosomal material resulting in extra or missing genes. Transpositions describe a segment of DNA sequence that move around the genome. And finally, splice site mutations, which we touched upon earlier, where mutations in certain sequences can result in aberrant splicing of introns. Oncogenesis is the process of acquiring multiple genetic alterations resulting in a disease state, namely cancer. Remember that epigenetic changes may also contribute to cancer formation, and this area of study continues to grow. Inactivating mutations in tumor suppressor genes or activating mutations in oncogenes are classical examples. Proto-oncogenes are normal genes in the body. A mutated proto-oncogene results in an oncogene, which promotes tumor growth. These are often the targets of cancer therapy drugs, as these genes tend to be potent driver mutations. The ultimate outcome of an oncogene is increased proliferation, decreased differentiation, and inhibiting normal cell death mechanisms. Only 1 copy of a proto-oncogene needs to be mutated to cause cancer. Tumor suppressor genes are genes that prevent tumorigenesis by coding for housekeeping processes within the cell, such as apoptosis, DNA repair, and cell cycle repression. The loss of a tumor suppressor gene results in cancer, and one of the most famous tumor suppressor genes is p53. For tumor suppressor genes, usually both copies are required to be mutated to induce cancer, and this is known as the 2-hit hypothesis, where 1 copy of the tumor suppressor gene is mutated and later acquires a mutation in the second copy, rendering the product of the tumor suppressor gene non-functional and allowing for cancer to grow. Let us briefly touch upon some of the abbreviations and common nomenclature rules, and then we will build on these further. DNA sequences are written in uppercase form. RNA sequences are lowercase. Proteins are often abbreviated into 3-letter or 1-letter forms, and there is a chart on the next slide to help you sort that out for your references. And then there are a bunch of prefixes and suffixes that describe the various components of genetic material and products. Some of the more common ones are described here. cDNA, coding or complementary DNA. gDNA, genomic DNA. MT stands for mitochondrial. MI for micro. DS stands for double-stranded. M for messenger. tRNA for transfer RNA, rRNA, ribosomal RNA, RNAi to describe RNA interference, snRNA, small nuclear RNA, nRNA for non-coding RNA, and then we have certain letters for designating genetic material or their products. R is short for RNA, P for protein, C for coding DNA, G for genome. This table lists the 3-letter and 1-letter codes for proteins, in case you wish to refer to this at a later date. There is a standardized nomenclature guideline for genes, known as the Hugo Gene Nomenclature Committee, or HGNC. Some of the more commonly encountered and useful rules to know include using italics for gene names and normal font for protein names to help distinguish these two in texts. Wild-type genes are designated with a superscript plus sign, while mutated genes will have a negative sign. Many other rules exist, and if you are interested, you can check out their guidelines at the link provided. Protein nomenclature is governed by the International Protein Nomenclature Guidelines, or IPNG. These provide a standard for naming and using proteins. Some useful rules include avoiding abbreviations, punctuation, plurals, or diacritics in naming proteins, the use of Arabic rather than Roman numerals, and enzyme names commonly end with ACE. See their publication in the link provided for a complete overview of the naming guidelines. When things go wrong in the DNA, RNA, or protein sequence, we use nomenclature rules established by the Human Genome Variation Society, or HGVS. These are a set of international standards that can be recognized no matter where a report is read. I encourage you to use their website to help you find or interpret annotation rules you may come across. Let us look at some of the symbols used to designate alterations. If there is a substitution, we use the greater than symbol, designating the original nucleotide or amino acid and their replacement. Nonsense mutations resulting in a premature stop are designated with the asterisk. Duplications use the symbol DUP, followed by the size of the duplicated sequence. Deletions use the abbreviation DEL, indicating the point or segment of DNA, RNA, or protein sequence that is deleted. Note that in-frame shifts are also represented by the DEL symbol. INS reveals the insertion sequence and the location of its insertion. Inversion is a segment of a sequence that flips around and reinserts itself in the opposite direction, and we designate these with the location of the inverted sequence followed by INV. Conversion describes a segment of DNA that takes on the sequence of another segment of DNA. The designation includes the region of the original segment of DNA and the location of the converted segment separated by the con symbol. Remember that a conversion is not a transposition. Conversion is a copy-paste, while transposition is a cut and paste. Frame shifts are designated with the lowercase fs, and will often tell you how long the segment of the shifted code is present and whether it ends in a stop codon or it may designate the new amino acid. So with any mutation or alteration, recognize that there are at least 3 different ways to describe it using nomenclature rules. Take these examples. A mutation in the coding DNA at position 1799 where T is replaced by A. A protein sequence where the 600th amino acid, valine, is replaced by glutamine. Or by using the name of the protein, in this case BRAF, followed by the mutation shorthand V600E. Notice BRAF is not italicized because we are referring to the protein name in this instance. All 3 of these represent the same thing, the BRAF mutation that is a common driver in numerous tumor types. So all these rules we've covered deal with the genetics and proteomics nomenclature. When it comes to the larger structures of DNA at the genomics level, as in chromosomal and karyotyping, The International System for Human Cytogenetics nomenclature has a set of international guidelines to follow. Let's begin by talking about how we locate genes or specific loci in the genome. Locating genes relies on the banding pattern of chromosomes. The string of words are organized starting from the chromosome number to the chromosome arm, the region, and then the banding position of the gene or locus of interest. So in the case of 14q22, this describes a locus on the 14th chromosome on the long arm or Q arm at region 2 in band 2. It is important to note that the phrasing of band and region are not combined, since these numbers represent different things. So we do not say 14q22, rather we say 14q22. The other way makes you sound like you don't know what you're talking about, and now you know better, so separate the bands and regions in your phrasing. The centromere is numbered 1O, with P10 facing the short arm and Q10 facing the long arm. Regions and bands are numbered from the centromere outward on each arm, so region 2 is closer to the centromere than region 3, and so on. With finer resolution, you may see an additional number following a decimal point, indicating a subband. The symbols CEN and TER tell you that the locus of interest is located near the centromere or terminal end, respectively. So in the case of 16p ter, we are referring to a locus on the short arm of chromosome 16 near the terminal end. Graphically, if we were to locate the CFTR gene, which is mutated in cystic fibrosis, we would look at chromosome 7. On the long arm, in region 3, band 1, subband 2. Here are some other symbols in cytogenetics that are useful to know. We use the plus and minus signs to designate the addition or absence of chromosomes, such as -7 or +21, to say a missing chromosome 7 or extra chromosome 21. If there are more than one clone present, we separate the two karyotypes by a single slash. In this case, there were two clones, one with an extra X chromosome and the other with normal karyotypes. We always list the normal karyotype last. Sometimes there may be more than two clones present, in which case we list the longest abnormal karyotype first, then the second longest, and if there are normal clones present, they will go last again. The numbers placed in brackets at the end of the karyotype tell us how many cells were identified with that karyotype. In this case, 10 cells were identified for each clone. In the case of post-transplant karyotypes, we use the double slash, with the recipient karyotype first, followed by the donor. In the first example, there are 3 male recipient cells present, with 17 donor female cells. The second example shows a complete transplant with all 20 cells showing a normal donor male karyotype. Translocations use the lowercase t followed by the chromosome numbers involved within parentheses and separated by a semicolon. This is often followed up by the location of the breakpoints for each chromosome. Translocations can be balanced or unbalanced. Balanced means there is an even exchange of material, so no extra or missing genetic information. These can be asymptomatic, unless the translocation results in a fusion protein like the 922 translocation, for example, which results in a fusion protein BCR-ABL1. Unbalanced translocations occur when the exchange of chromosome material is unequal and results in extra or missing genes. These are generally more sinister, as you can imagine. Marker chromosomes are incomplete fragments of a chromosome that lack a centromere and are unidentifiable. Derivative chromosomes have more than one alteration in them, such as an inversion and a deletion, or an unbalanced translocation, for example. They are designated based on the centromeric region they have retained. Incomplete karyotypes use the INC abbreviation at the end of the karyotype string. It is important not to overlook this designation, as it signals that there may be more unidentified structural or numerical changes in addition to the abnormalities that are listed. Without this designation, one may conclude that only the abnormalities listed are present, when in fact there are other aberrations that may exist that could not be accounted for due to the poor quality of the specimen. When we find aberrations and report cytogenetic findings, we combine everything into a final string using a standard order, starting with the overall number of chromosomes, the sex chromosomes, affected chromosomes, type of abnormality in shorthand, and number of cells with the given karyotype in brackets. Many of the symbols and annotations used to describe the aberrations in cytogenetics are shared with those we looked at previously under HGVS guidelines. When describing the abnormalities in a karyotype, there is an order to them as well. Sex chromosomes come before autosomes, with X abnormalities coming before Y. Autosomes are ordered in numerical order. For each chromosome, numerical abnormalities come first, followed by structural changes. Multiple structural changes within a chromosome are listed in alphabetical order. Here are some examples of this order in action. The first example, a female karyotype without aneuploidy, 46XX, but this specimen shows a 922 translocation, also known as the Philadelphia chromosome. The 9th chromosome, at position Q13, has been translocated to the 22nd chromosome at position Q22, which results in the abnormal BCR-ABL1 fusion protein often seen in leukemia. And this abnormality was found in 20 cells. 20 is the typical number of cells reported in karyotypes, unless there aren't enough cells to evaluate, of course. The second example describes a total number of 47 chromosomes in a male patient as a result of an extra copy of chromosome 21, and this was seen in 12 cells, indicating there likely were less than 20 cells available for evaluation. The third example describes 18 cells showing a loss of the long arm of chromosome 5 at region 3, band 1, and 2 cells showing a normal male karyotype. This abnormality is seen in myelodysplastic syndrome. And finally, in the last example, there is an inversion in chromosome 16 in this female patient, an abnormality seen in cases of acute myeloid leukemia. All these examples are very straightforward. But oftentimes, tumors will have very complex cytogenetics. Reports may show a karyotype filling an entire line or even filling a paragraph, like the last example here from a sarcoma. In these instances, if you wish to decode the entire cytogenetic profile, you will probably need to refer to the ISCN nomenclature publication. And in case you were wondering, question marks are in fact part of the nomenclature, representing a questionable or unknown identification. In clinical practice, a summary report may accompany the karyotype to highlight key prognostic or therapeutic findings, but generally, if a karyotype is this long, you know it's not a good sign. ISCN also regulates fluorescent in situ hybridization nomenclature, which has a set of its own nomenclature rules, some of which is less intuitive to a beginner, but I will try to simplify as much as I can as we go through some of the more common rules. When we describe a fusion gene in text, we use the hyphen between the two genes. Notice these are italicized as we are describing the genes and not the proteins. When describing fish probe cocktails, we use the slash instead. We identify the locus targeted by fish probes by the clone name of the locus. If that is not available, then we assign the name based on genome browser databases, either Ensemble Gene Browser or UCSC Genome Browser. If neither of those are available, we use the D number for the probe. I will not be digging too far into the weeds on how these probes are named. In the clinical setting, FISH probes are generally designated by the gene they target. If FISH is to be reported alongside karyotype results, we report the karyotype first, followed by the FISH results after, and separate the findings with a period. Oftentimes, though, two separate reports are issued to avoid any confusion by the clinicians. If in situ hybridization occurs in metaphase or prophase cells, we use the lowercase prefix ish, ish. If in situ hybridization occurs in interphase cells, we use the prefix nuc-ish, nuc-ish. One way to tell the difference by looking at the screenshot of the FISH analysis is that ish will show DAPI staining in compact chromatids, while nuc-ish will show an entire DAPI-stained nucleus as a sphere. We follow these prefixes with a space. Here are some examples of FISH reports. In the first example, a normal karyotype is shown, separated by a period, from the FISH results. FISH was performed on metaphase cells, as designated by the ish prefix. The probes used targeted the X centromere, labeled by the D number DXZ1, and the SRY gene located on the Y chromosome. Two signals of DXZ1 probe are seen, and no signals of the SRY probe are seen, confirming a normal female karyotype. The second example tells us there is an extra marker chromosome identified on karyotype. FISH analysis on metaphase cells identify the marker chromosome to be derived from chromosome 8 using the 8-specific probe called D8Z1, showing an extra signal. Since we have identified this marker chromosome, we use the designation DER to show that this is a derivative 8 chromosome and is no longer unidentified. In the third example, no karyotype is available. We are only looking at the FISH results performed on metaphase cells, using the probe against RUNX1 gene. The results showed an amplified signal in 4 cells that cannot be enumerated. If the number of amplifications can be counted, then they would have designated the number of signals. This abnormality can be seen in some leukemias. The fourth example shows an interphase FISH result showing amplification of mixed signals, between 12 to more than 50 copies in 200 cells evaluated. And in the last example, FISH on interphase cells showed a lost signal for the ATM probe in 90 of the 200 cells analyzed. TP53 was intact, with 2 signals present. The remaining 110 cells are inferred to be normal for ATM. In FISH, the relative position of the probes makes a difference, and this information needs to be conveyed as well. If loci on 2 separate chromosomes are tested, the signals are expected to be spatially separated. In this example of 100 interphase cells, 2 signals are seen for ABL1 and 2 signals for BCR probes. If a 922 translocation occurs, then one BCR signal comes close to one ABL signal as a result of forming the BCR-ABL1 fusion protein. This is conveyed by a first set of parentheses showing the total number of signals for each probe. In this case, 2 signals were seen for each BCR and ABL1. Then a second set of parentheses describes the relative position of the signals to one another. In this case, con is used to show that one BCR probe converged or is juxtaposed with one signal of ABL1, resulting in a yellow signal. The same rules apply with dual fusion probe. In this case, we see only 3 signals each for ABL1 and BCR probes, and this is displayed in the first set of parentheses. The second parenthesis shows that two fusion signals are present between the two probes. The difference between single fusion and dual fusion probes has to do with the way the probe is designed around the breakpoint of these genes. Single fusion probes do not span the breakpoint of the gene, so if the gene breaks and translocates elsewhere, you still only get one signal for the gene on each chromosome. A dual fusion probe does span the breakpoint, so when the gene breaks, there are two signals, one on each half of the broken gene. The third signal comes from the intact probe on the unaffected chromosome. We will discuss signaling patterns in more detail in Part 2. In the case of break-apart probes, the normal signal pattern is two yellow signals. In these cases, since the probe spans the breakpoint of a gene, we need to designate the 5' and 3' ends of the probe when it breaks. Take for example this probe of the MYC gene, with two normal signals. We can use the short form nomenclature to show that there are two MYC signals. The long form nomenclature shows that the 5' and 3' ends of the break-apart probe are still in contact, using the con designator. Both of these examples are acceptable for normal in this case. When a break occurs, we now have to designate the separation of the signal using the SEP designation. In this example, there are two MYC probe sets present, as demonstrated in the first parentheses. The second parentheses tells us that one of them shows a separation of the 5' and 3' ends of the break-apart probe. Again, we will discuss break-apart probes in more detail in Part 2. And finally, I will quickly mention microarrays, but we will only dip our toes in for completion's sake. A microarray is designated by the ARR prefix. A normal result lists the sex chromosomes first, followed by the autosomes, as shown here for male and female samples. If the results are abnormal, then only the abnormalities are listed. When defining an abnormality other than aneuploidy, the genome build used as reference should be listed as well. An example of this is shown here. The Genome Reference Consortium Human Build 38, or GRCH38, is listed in brackets, telling us the reference sequence used in the microarray. In the first abnormal example, the CX designation indicates a complex microarray result across a female genome. In the second example, the microarray analysis shows an interstitial loss in the long arm of chromosome 6 at bands Q21 through Q25.1. The large numbers separated by an underscore in the parentheses are the location of the aberrant nucleotides as they would be found in a genome database, such as Ensemble or UCSC Genome Browser. In addition to the segmental loss in 6q, there is a single copy gain, or trisomy, in chromosome 21. SNP arrays provide additional information, such as homozygous and heterozygous tracts, and this information is added to the report, as shown in the last example. In this short-form SNP array analysis, there is homozygosity for the entire long arm of chromosomes 15 and 21, and both regions of homozygosity are obtained from the father. The abbreviations HMZ is used for homozygous, HTZ for heterozygous, MAT for maternal, and PAT for paternal. We will discuss microarrays in more detail in Part 2. Okay, that was a lot of material covered, but I hope you found this review useful.